Good evening, dear students. So, we are discussing on the topic biomolecules, right? So, we have completed carbohydrates in our previous class. So, we have discussed all so we have taken different examples of sugars for monosaccharide, disaccharides, and polysaccharides, right? So, we have completed carbohydrates in our previous class. Then we had started with uh, yes, so we were discussing on amino acids. Yes, so what are amino acids? What are amino acids? So we have discussed about amino acids, the 20 amino acids, right? Then we have seen the classification of amino acids. What are amino acids? Yes, so they are the compounds which are containing the both amino group as well as the carboxylic acid group. Okay, in the same molecule, in the same compound, both the groups are present. That is one acidic group and one basic group is present, right? So amino group. Then we have seen 20 types, 20 naturally occurring alpha amino acids, right? So how they are categorized? Yes. So we have seen the classification also of amino acids. So based on the nature of amino acids, they are classified into three types. Okay. Acidic amino acid, basic amino acid, and neutral amino acid. So we have discussed this also. What are acidic, what are basic, and what are neutral amino acids. Then on the biological importance, based on the biological importance, okay, they are classified as essential amino acids and non-essential amino acids, right? So what are essential amino acids? Which cannot be synthesized by our body and it should be taken through diet. Okay. So such amino acids are called as essential amino acids. And what are non-essential amino acids? So these are the amino acids which can be synthesized by our body. Okay. And so these are synthesized in our body. So these are called as what? Non-essential amino acids. Okay. So this was about amino acid. Then we had discussed about something called as isoelectronic point and Zwitter ion, right? So what is this isoelectronic point? Yes. So it is a pH of the amino acid. Okay. So at that particular pH, the amino acid will not move either to the anode side or it will not move to the cathode side. So that particular point or that particular pH of the amino acid, okay, in the aqueous solution, it is called as what? Isoelectronic point. Okay. Then what is Zwitter ion? Yes. What is Zwitter ion? What is Zwitter ion? So we had seen the structure of alpha amino acids, right? We had seen the structure of alpha amino acids. So you can see that the carboxylic group, okay, so the carboxylic group is having a hydrogen, that is a proton is present, right? So this proton can be easily donated to the amino group, right? So the hydrogen, that is a proton from the carboxylic group, it can be easily, it can donate, okay, donate a proton to the amino group and thus a dipolar ion is formed and this is called as Zwitter ion. Okay, so this condition it undergoes in a aqueous solution. So when amino acid are, okay, when we take it in the aqueous solution, so that time this process takes place. So the proton is donated, okay, from the carboxylic group to the amino group, okay, and it forms a dipolar ion and this is called as what? Zwitter ion. Fine. Okay. Yes, very good. Sri Varuni has answered. Very good. Okay. So let us go ahead. Okay, let us move ahead. So today we are going to discuss the next part. So next we are going to see the peptide formation. Okay, peptides and polypeptides. Now, what do you mean by the word peptides and polypeptides? Okay. So what do you mean by the word peptides and polypeptides? Now, first we will see peptide formation. Okay, how this peptide formation takes place. So, a amide bond. How is a amide bond? That is C double bond O NH. Okay, C double bond O NH bond is called as what? It is a amide bond. So this amide bond, it is formed between two amino acids. Okay, between two amino acid. Okay, so suppose now I have A and I have B. I want to make them together. I want to make them together. Then what I'm going to do? I'm going to add them. Right? I'm going to add them. So if I add, it will become AB. Right? It will become AB. Similarly, now I have two amino acids. Okay. I have two amino acids. Now this amino acids, they should be joined together. Right? They are joined together. So when these alpha amino acids, okay, when they are joined together, they are joined through a bond. Okay, they are joined through a bond. And in between these two molecules, okay, between these two alpha amino, between these two amino acid, okay, what will happen? There will be elimination of water molecule. Now here you can see, this is one amino acid, this is another amino acid. Okay, now what is happening? So when the two amino acids are okay joined together, there will be formation of a CONH bond. This is CO and this is NH bond. That is your amide bond with the elimination of water molecule. Okay, with the elimination of water molecule, and that bond formation is called as a peptide bond formation. Okay, so when two amino acids, okay, when they are added, okay, what is formed? A amide bond is formed between these two amino acids, okay, acid. And what happens with that? There will be elimination of water molecule. And the bond which is formed, it is called as the peptide bond or peptide linkage, okay. So this is about peptide formation. Understood? Is it clear? Suppose, okay, so let me take one example here. So I'll write it down. Okay. Now suppose I have NH2, CH2, okay, C double bond O, OH. So this is one amino acid. This is a glycine. Okay, glycine. Again one, I'll take amino acid. Let me take aniline. Okay, let me take aniline. So this is NH2. Okay, then I have CH, CH and COH. Okay, so this is your aniline. Okay, aniline. Fine. Now here what happens when these two are added? Okay, when these two are added, what will happen? There will be formation of what? There will be formation of a amide bond between them with the elimination of water molecule. So this is the water molecule H2O. And what is the bond formed? How it is formed? So NH2, okay, CH2. Then is what? C double bond O. Then is NH. Then is CH, CH3 and COOH. Okay, understood? So between these two amino acids, okay, they are linked by a peptide bond. Okay, that is your amide bond. Okay, with the elimination of water molecule. Is it clear? What is peptide bond? Yes, is it clear? Very important. What is peptide bond? Okay. Now, what do you mean by dipeptide? Okay, now what do you mean by dipeptide? Okay, dipeptide means what? Dipeptide consists of two amino acids. Okay, it consists of two amino acids. They are held together by one peptide bond. Okay, one peptide bond is formed. So dipeptide means what? In a dipeptide, two amino acids are present and one bond is formed. Now you can see here. Now these are two amino acids and only one bond was formed. So this is only one peptide bond is formed in between the dipeptide. Okay. Similarly, if I say tripeptide, in tripeptide, how many bond will be formed? Yes. How many bonds will be formed? Yes. Now in dipeptide, one peptide bond is formed. So in tripeptide, how many bond will be formed? Two. Yes, very good. Baby has answered. Very good. So it is two bonds are formed. Okay. So remember, like this, the question is asked. Okay. What is a peptide bond and how many bonds are present in a tripeptide? So you have to answer two peptide bond are present. Okay. Now in a tetrapeptide, yes, tetrapeptide, three bonds are formed. Okay. So similarly, similarly, what is happening here? So the number of peptide bonds. Okay. So the number of peptide bonds in di, tri, and tetra. So if it is a dipeptide, okay. So how many bonds are present? One. If it is a tripeptide, how many number of peptide bonds are present? Two. Okay. If it is a tetrapeptide, okay, how many number of bonds are present? Three. Okay. If it is a pentapeptide, then it is how many bonds are present? Peptide bond, four. Okay. So this is, you have to remember. So this is peptide formation. Next, we are going to study about polypeptide. Okay. So next, we are going to study about polypeptides. Now, what do you mean by polypeptide? Poly means what? Many. Poly means many. Okay. So when more than 10 amino acids are there, okay, when more than 10 amino acids, okay, when they undergo condensation, okay, when they undergo condensation, okay, then the product formed will be a polypeptide. What it is formed is polypeptide. So when it is more than 10, so when there are more than 10 amino acids, Okay, and if they undergo condensation, then what is formed? Polypeptides are formed. Okay, polypeptides are formed. So this is about polypeptides. So next we are going to start with proteins. Okay, next we are going to start with what? Proteins. So when you have a polypeptide, now you know what is polypeptide. Polypeptide means when you have more than 10 amino acids, then polypeptide is formed. Okay, so a polypeptide with more than 100 amino acids. Okay, so when you have a polypeptide, okay, when you have a polypeptide and which is having more than 100 amino acids, okay, then they are having molar mass also will be more, okay, their weight also will be more, so the weight will be more than 10,000 unified mass, okay, then they are called as protein. Then they are called as what? Protein. Okay, so what, so when you have a polypeptides and if you have more than 100 amino acids, okay, so when a polypeptide with more than 100 amino acids, okay, whose mass, okay, whose mass or molar mass is more than 10,000 unified mass, then they are called as what? They are called as protein. Okay, they are called as what? Protein. So based on the molecular shape, so based on the molecular shape, they are classified as two types. Okay, they are classified as two types. First, we will discuss the types. Okay, then we will go for the this what structure of protein. So before that, so what is protein we have discussed. So when you have a polypeptide with more than 100 amino acids with the molar mass, okay, with the molar mass more than 10,000 unified mass, then they are called as protein. Okay, so based upon the molecular shape, so based on the molecular shape, they are classified as fibrous proteins. Okay, they are classified as what? Fibrous proteins and globular proteins. Okay, fibrous proteins and globular proteins. So there can be a question. What are fibrous proteins? Okay, give example. What are globular proteins? Give example. So 2 marks question. 2 marks question. Mark is important. What is fibrous protein? Give example. Uh, what is globular protein? Give example. So first, let us discuss about fibrous proteins. Okay. So in fibrous proteins, you have long parallel. Okay. You have long parallel polypeptide chains. Okay. You have long parallel polypeptide chains. So, just like your threads, okay, just like the threads, we have the polypeptide chains. So, like this. So, this is the your fibrous proteins. Okay, so they are long, they are parallel, they are okay with a straight like chains, okay, and like threads, and they are held together by hydrogen bond. So, what bond is present? Hydrogen bond is present and disulfide bond. Okay, and disulfide bond. So remember this. So what is bonds are present? Hydrogen bonds and disulfide bond. Disulfide bond to form fibers. Okay, to form fiber-like structure. Fiber-like structure. So remember this. So what type of bonds are present? Hydrogen bonds is present and the disulfide bonds are present. Why disulfide bonds are present? So that they form fiber-like structure. Okay. And these your fibrous proteins, they are generally insoluble in water. Okay. They are insoluble in water. So what are the examples of fibrous proteins? Okay. Your keratin. Keratin, where it is present, students? Keratin, it is present in hair. Okay, keratin is present in hair. Okay, so if more the keratin, if more the keratin, your hair will be straight like that, right? So keratin, it will give you a smoothie texture. So keratin, it is present in hair. Then it is present in wool. Okay, then silk. Okay, then another is myosin. Okay, myosin, it is present in what? It is present in muscles. So these are the two fibrous proteins. Okay, these are the two fibrous proteins. So what are fibrous proteins? They are the long parallel polypeptide chains, okay, which are held by the hydrogen bonds and disulfide bonds to form fiber-like structures. These are thread-like structures, okay, and they are insoluble in water. What is the example? Keratin, which is present in the hair, okay. Then it is present in wool and silk. Then myosin, it is present in the muscles, okay, in the muscles in our body. So these are the fibrous protein. Next is globular protein. Next is what? Globular protein. Now, what are these globular proteins? These are coiled, okay. So how these were straight? These are coiled. These are coil-like structure. So these proteins have polypeptide chains. So these are also having polypeptide chains, but these polypeptide chains are coiled, okay, around to give you a spherical shape. So it is giving you a spherical shape. So it is coiled, okay, it is forming a coil-like structure. And these are usually soluble in water. Okay, these are soluble in water. What is the example? Example is albumin. Please note down this. Okay, and insulin. Insulin it is present in the what? It helps in to regulate the blood sugar, right? Insulin, albumin. Okay. So these are the globular proteins. Okay. So what is fibrous proteins and what is globular proteins with example? Is it clear? Is it clear? Okay, if you have any doubts, you can ask. Okay. So what are fibrous protein? Fibrous proteins, they are long parallel polypeptide chains, okay, which are thread-like structures, and they are held by hydrogen and disulfide bonds to form fiber-like structure. These are insoluble in water. Example, keratin and myosin. Keratin, it is present in hair. Myosin, it is present in muscles. Globular proteins, these are proteins which are having polypeptide chains which are coiled in a spherical shape, and these are soluble in water, which are the example albumin and insulin. Okay. Now next we will see the structure of protein. Next is the structure of protein. So next is the structure of protein. So the structure of protein is, we are studying the structure of this word protein at four different levels. That is primary, secondary, tertiary, and quaternary. So we are studying the structure of this at four different levels. Okay. So primary, secondary, tertiary, and quaternary. So let us see at each this, okay, at four different level, how is the structure of protein given. So first we will see primary. Okay, so primary, what it will it will specify. Okay, it describes the specific sequence of amino acids. Okay, it specifies what it describes the sequence. Okay, how the amino acids are here, how it is sequence. Okay, so how in which sequence the amino acids are added. Okay, so that sequence, it is decided by the primary. Okay, so primary structure of protein, what it refers to the sequence of amino acids in each polypeptide chain. So we know proteins, they are made up of long polypeptide chains, right? Now these polypeptide chains, how they have arranged, okay, how the sequence is given. So that is decided by the primary structure of protein. So any change in the primary structure means there will be change in sequence. So if there will be change in sequence. So if any change occurs in the primary structure, means there will be change in the sequence of amino acid. And because of the change in sequence of amino acid, what will happen? Different protein is created. Okay, it creates different protein. So this is about primary. So what you have to remember about primary structure of protein, it it uh, it specifically describes the sequence of amino acids. Okay, so if any change in the primary structure of amino acid, what will happen? There will be change in sequence of amino acid and a different protein is produced. Okay, so this is about primary structure of protein. Okay, this is about primary structure of protein. Secondary, next we will see secondary structure. So this secondary structure, it tells you about the shape. Okay, it tells you about the shape of long polypeptide chains. So it decides the shapes. Okay, it decides the shapes of the polypeptide, long polypeptide chain. So the chain can be existed in two different forms. One is alpha helix and another is beta pleated sheet. Okay, collagen helix is not there for you. Only these two. So the secondary protein, okay, structure of protein, what it tells you, what it describes the shape, okay, of the long polypeptide chain. So alpha helix means like this. So helical form. So this shape, okay, if the proteins, they are coded like this in helix form, then it is called as the alpha helix. The shape is called as alpha helix. And beta pleated means what? See, in the plates form, okay, in the form of plates. So this is a beta pleated sheets, okay, in the form of sheets. So this is in the form of plates and with the thin sheets like this. So these are the two what's shapes, okay, of this tells you the shape of the secondary protein. Okay, next one, tertiary. Okay, what does tertiary tells you? It tells you about the region of secondary structure. Okay, and it tells you, so so it describes how the region of secondary structures fold together to get a 3D arrangement of polypeptide chain. Okay, so it gives you two different molecular shapes for protein, that is fibrous and globular. Okay, so how this secondary, that is your your beta sheeted pleat, okay, you can see. So how it will undergo, okay, the transformation into a 3D structure. So this is your primary structure, this is your secondary structure, this is your tertiary structure. Okay, so what we are doing in primary we are arranging in a sequence. Okay, and in secondary what we are doing, they are deciding the shape. Okay, either if it is arranged in alpha helix form or in a beta pleated shape. And tertiary, what it tells you, it together. Okay, so how the arrangement, okay, how the sequence of your amino acids, they are arranged in the shape, okay, to give you a 3D structure or 3D arrangement of polypeptide chain. So it represents the overall foldings, okay, how the foldings here, how it is given, this particular structure, last structure, right? So together, so first and second together, it will give you a tertiary structure. That is overall foldings of the polypeptide chain, okay. And two types of this majorly it will give you, that is this is bit complex. Okay, so the structure of tertiary is bit complex because it involves the 3D structure. Okay, so secondary and tertiary structures of proteins are stabilized. How? In that hydrogen bonds are present. Okay, hydrogen bonds are present, disulfide linkage is present, van der Waals forces are present, electrostatic forces of attractions are present in primary and secondary structure. In tertiary, okay, so tertiary is bit unstabilized, okay, because it is a 3D structure. Fine. Next is quaternary structure of proteins. Next is what? Quaternary structure of protein. Now, what does quaternary structure? So, some of the proteins, they are said to be quaternary, okay, if they have two or more polypeptide chains, okay. So, here your hemoglobin is the best example of a quaternary ammonia, that is quaternary chain. Okay, your quaternary protein. Fine. So here the spatial arrangement. Now here you can see. So what will happen? Two or more polypeptide chains, they will give you different spatial arrangement. So this is the alpha helix form. This is a beta change. Means what? This is one polypeptide chain, this is another polypeptide chain, this is another polypeptide chain. Okay, and this is what? This is a heme protein. Heme protein means it is a hemoglobin protein. Okay, so when two or more polypeptide chains are present, so that will give you a quaternary structure. Okay, so this is about the structure of protein, very important. Okay, so if they ask you to explain the structure of protein, you have to explain primary structure, secondary, tertiary, and quaternary. Okay, so this will, so here you can see, so primary. Okay, so the arrangement, the sequence of different amino acids in a proper sequence, then what will happen? These sequence it will be sequenced. Okay, and then it will undergo a helix form. Okay, so slowly it will start to coil. So this is your secondary structure. Okay, it undergoes a secondary structure. So how the you are adding the amino acids, like that, the it will start to in the sequence it will start to coil. Okay, and tertiary, what will happen to tertiary? So these different amino acids. Okay, so when it is arranging, okay, what will happen? So it will give you a tertiary, that is a 3D structure. So this is your tertiary structure of protein. Quaternary, in quaternary, there are two or more polypeptide chain. Okay, and they will give rise to a quaternary structure. Okay, so this is about the proteins. Okay, this is about the proteins. Now, where these proteins are important, proteins, they function as enzymes. Okay, they help. They are what in the biological system. Okay, they perform like antibodies. Okay, then they are used as the transport agent. Okay, for transporting. Then they are used as a biochemical messenger, that is hormone. So these are some of the functions of the protein. Okay, fine. So here you can see. So different functions. So structural protein. Okay. So keratin, collagen. So this is present in the skin. Collagen is present, and in the hair, keratin is present. So it will give you the structure for the hair. Collagen will give you structure for the skin, face. Enzymatic protein, pepsin. Okay, so it is an enzymatic protein, then which is the transport protein of our body, hemoglobin. Then hormonal proteins, insulin, growth hormone. Then contractile protein, actin and myosin. Okay, so then storage protein, olalbumin. Okay, then genetic protein, nucleoproteins. Defense protein, immunoglobulins. And receptor proteins for hormones and viruses. So these are the functional classification. Then depending upon the okay, chemical nature and solubility, they are classified as simple proteins, conjugated and derived. Simple means they are composed of only amino acids. Conjugated means along with amino acid there will be non protein prosthetic group. So other different group will be associated with the amino acid, then they are called as conjugated protein. And derived means they are denatured or degraded products of the above two. Okay, so from simple and conjugated, we are going to derive proteins. Okay, so they are man-made like proteins. So this is about your proteins. So next we are going to discuss about the enzymes. Okay, what are enzymes? So these are the globular proteins which are produced by living organism. Okay, so what are enzymes? So note down about enzymes. So what are enzymes? So enzymes, these are the globular proteins, and they are produced by whom? They are produced by living organism. Okay, and what they will do? They will catalyze the biochemical reaction. What they will do? They will catalyze the biochemical reaction. So this is the function of your enzyme. Okay. So enzymes, they are produced by living organism, and what they do, they catalyze the biochemical reaction. Okay. Then these enzymes are very specific. Okay. So they are very specific at so they are very specific, and they depend upon the particular temperature and pH of the solution. Now, which is the different enzymes. Now to convert the maltose, okay, to convert the maltose into glucose and glucose. Okay, so maltose, when it undergoes hydrolysis, it will give you what? 2 moles of glucose. So if you use the enzyme maltase, okay, it will give you 2 moles of glucose. So these are the enzymes. So what are enzymes? Just remember the definition. So enzymes, they are the globular proteins which are produced by living organism to catalyze the biochemical reaction. Okay. Next is vitamins. Next is what? Vitamins. Okay. Now what are these vitamins? These are the organic uh, compound which is required in diet. Okay, and in very small amounts, so that in the body any biochemical functions can be carried out, right? So that our body can it can be maintained, right? To have a proper diet, so we should have a proper diet, so that whatever we are having, so it should include vitamins. Okay, and this vitamin you should take it, means it should be taken in the proper proportion. Okay, so that the human body or the living organism can have a normal means growth, and it can be maintained. Okay, the health can be maintained. Now what is the classification of vitamins? What is the classification of vitamins? So depending upon their solubility, so depending upon their solubility, okay, so the vitamins, they are classified as fat soluble, okay, fat soluble vitamins, and water soluble vitamins. Okay, they are called as fat soluble vitamins or water soluble vitamins. Okay, so now what is fat soluble vitamins? Fat soluble means these vitamins are soluble in fat. Okay, fats and oils. They are soluble in fats and oils, but they are insoluble in water. Okay, they are insoluble in what? They are insoluble in water. Okay, and where they are stored in liver. Okay, in our body, liver is there, right? So there, these are stored. So they are stored in liver and adipose tissue, and adipose tissue. So these are the fat soluble vitamins. So the vitamins which are soluble in fats and oils, okay, but insoluble in water, they are called as fat soluble vitamins. Where they are stored, stored in liver and adipose tissues. So please remember, very important 2 marks question. Again, mark this important 2 marks questions. What are fat soluble vitamins? Give example. What are water soluble vitamins? Give example. So what is the example for fat soluble vitamins? Vitamin A, D, E, and K. Okay, vitamin A, D, E, and K. Now what is water soluble vitamins? Okay, so these are soluble in water. Okay, these are soluble in water, and they are supplied in regular diet. Okay, and these cannot be stored in body. Water soluble vitamins, they are soluble in water, so we have to take it regularly in the diet. Okay, because it will go away from our body, it will readily get excreted, because it cannot stay in the body, it cannot be stored in the body. Why? Because it is water soluble. We drink water, right? Whatever we, if they are water soluble, what will happen if we drink? Okay, so after when we urinate, what will happen? The vitamins will get what? They will flow out, right? So because they are soluble in water, these vitamins, it should be taken in our regular diet. Okay, it should be taken in our regular diet, and they cannot be stored in the body. Now, which is the example? Which is the example? Vitamin B group. Okay, all the vitamin B group. So these are the what? Water soluble vitamins, except B12. Okay, except B12. All other vitamins, they are soluble in water. Okay, all other vitamins, they are soluble in water. And vitamin C, and vitamin C. Okay, so these are all soluble in water. Is it clear? What is what vitamins? What are the classification of vitamins? Yes. Okay. Fine. So next we are going to see the deficiency of vitamins. Okay, different vitamins have different deficiency. So let us see that. Just a minute. Okay. So you can go through this from your textbook also. So in the textbook there is a chart, okay, with which is the name of the vitamin. Okay, so name of the vitamin, then which are the sources of that vitamin, and what is the deficiency. Okay, what is the deficiency diseases which is caused when you have lack of that vitamin. So when you have vitamin A. Okay, vitamin A, the source of vitamin A is fish, liver oil, okay, carrots, butter, and milk. Okay, what is the deficiency? That is xerothalmia. Okay, so that is your eye. Okay, there will be hardening of the cornea of eye. So in the eye, what will happen? The shell, it will become hard. Okay, so because of the less amount of source of vitamin A, and also it causes night blindness. So in the night you cannot see. Okay, because of vitamin A. Then vitamin B1. So if vitamin B1, if there is deficiency in vitamin B1, it causes beriberi. What do you mean by beriberi? Beriberi means loss of appetite. You don't feel hungry. Okay, loss of appetite. So if there is a law, this vitamin B1 is less in your body, you will not feel hungry only. Okay, there will be loss of appetite. And what will happen? Your growth, you will not you will become very lean and lean. Okay, your growth won't be what you say, it will be retarded. Okay, your growth will be retarded. There won't be a proper growth. So if you have vitamin B1, so which are the sources of vitamin B1? Milk. Okay, green vegetables, so you have to have more milk, green vegetables, then all the cereals you should have. Okay, so if you have these sources, then vitamin B1 will be properly in your diet. Okay, then vitamin B2. Vitamin B2, okay, that is riboflavin. Okay, riboflavin. So this, what is the sources? Milk, okay, then egg white, then liver, okay, kidney. So these are the sources. Then what it causes? It causes digestive disorders and burning sensation of skin. Okay. So this is about vitamin B2. Then vitamin B6. Vitamin B6 convergence. Okay. Convergence caused by the vitamin B6. Then vitamin B12. Okay. What are the sources? Meat, fish, egg, and curd. Okay. What it causes? Pernicious anemia. Anemia means what? In the body, blood count will be less. Okay. It will become less and less because of vitamin B12. Then is vitamin C, that is ascorbic acid. So because of ascorbic acid, that is vitamin C. Okay. What will happen? Scurvy. Scurvy means what? Your below the teeth, gums will be there. No, from that gums, blood will start to come. Okay. There will be bleeding comes. It means scurvy. So what are the sources of vitamin C? So you should have more citrus fruits. Okay, citrus fruits like orange, lemon, Okay, so these are the citrus fruits. Then amla, you should have amla and green leafy vegetables. So if you take these sources, then vitamin B, you will take vitamin C. Then next is vitamin D. Okay, vitamin D, it causes rickets. So lack of vitamin D causes rickets. Okay, that is bone deformities in uh, children. So how, which is the sources to get vitamin D? Exposure to sunlight. So you have to stand in the sunlight, not in the hot sun at 11 o'clock. Okay, from 7 to 9. So in between 7 to 9, okay, AM in the morning. So that time, at least for 5 to 10 minutes, you have to stand in the hot sun. Okay, you should get exposed to the sunlight. So you will get vitamin D, natural vitamin D you will get. Then from fish, and egg yolk. Okay, egg yolk means yellow color. Okay, of the egg. So that is, so these are the vitamins and their deficiency. Okay, this is about the vitamins. Any doubts here? Yes, any doubts about the vitamins? So we are left with the last part, that is nucleic acid. Okay, we have left with a very small part, that is nucleic acid. So let us complete it today itself. Nucleic acids. So what are nucleic acids, students? Can anybody tell me? So nucleic acids, what are the composition of nucleic acids? Yes, what are the composition of nucleic acids? So what does this? Yes, RNA, DNA. Okay, fine. Now here, what are these nucleic acids? Okay, and why these are so important in our life? Okay, so why they are more important? Now we know that. Now see, now your dad. Okay, if you see, you will resemble okay yourself like your mom or like your dad. Okay, your dad, he will resemble like his dad or his mom, like that. So this is generation. Okay, all the generation. So they are having some unique this what features, right? So that features, like some people tell you, you look like your grandfather, you look like your grandmother, right? So there is some common thing between you, your ancestors, right? So the characters, how they are transmitted from one to the next generation. Okay, and how it is transmitted. Okay, uh, it is called as heredity. It is called as what? Heredity. Now suppose some of them you have seen, they have cat like eyes. Okay, blue color eyes. So that eyes, it will be seen in their kids also, like that. So what it is transferred. So some of the features, they are transferred to their kids. Okay, to their kids, like that. So that is what heredity. So this is responsible. So what is responsible for heredity? The chromosomes. Okay, chromosomes are responsible for the heredity. And these chromosomes, they are made up of proteins. Okay, they are made up of proteins and nucleic acids. Okay, so because of this, nucleic acids are very important. Okay, so because of this, nucleic acids are very important. So heredity. Okay, so from ancestors to ancestors, so from your forefathers, you will have some common features. Okay, in between you all the people. So in your family. So that is heredity. So heredity, it is transferred from chromosome. So chromosome, it is made up of what? Proteins and nucleic acid. Okay. So there are mainly two types of nucleic acid. That is deoxyribonucleic acid. That is DNA. Okay. That is deoxy ribonucleic acid. Okay, that is your DNA, and ribonucleic acid. Okay, and ribonucleic acid, that is RNA. Okay, it is RNA. So these are what? These are the long chain of polymers of nucleotides. Okay, so they are called as polynucleotides. What they are called as? Polynucleotides. So these DNA and RNA, they are made up of long chains of nucleotides. Okay. So they are called as polynucleotides. So let us see the composition. Okay. Composition of nucleic acid. Is it clear? Understood about the introduction of nucleic acid? Why nucleic acid are important? Okay. If anybody asks you why nucleic acids are important, will you be able to tell? Yes, why nucleic acids are important in our day-to-day life. Now you know why carbohydrates are important, why proteins are important, why vitamins are important, why enzymes are important, why hormones are important, right? So these are all important things which are required by our body so that the body will synthesize, okay, and it will be used for the growth and maintenance of the body, right? So now when we are discussing about nucleic acid, so let us discuss about the composition of nucleic acid. Okay, composition of nucleic acid. So let us see the composition of nucleic acids. So when nucleic acid, what it consists of? DNA and RNA. Okay, DNA and RNA. So nucleic acid, it consists of what? DNA and RNA. So this DNA and RNA, it breakdowns into different units. Okay. When we break down this DNA and RNA, it breakdowns into different units. Okay. How it break down into different units? Okay. So nucleic acid, in the presence of nuclease, okay, it gives nucleotides. Okay, it gives you what? Nucleotides. This nucleotides, okay, it forms nucleoside. Okay, nucleoside plus phosphoric acid. Okay, this nucleosides, in the presence of nucleosidase, okay, nucleosidase is the enzyme. It produces pentose sugar, okay, and nitrogen bases. So nitrogen bases and pentose sugar, these are very important. So the nucleic acid, they are made up of what? They are made up of pentose sugar and nitrogenous base. Okay. So if they ask you what is the composition of nucleic acid, so nucleic acid, it is made up of pentose sugar. So pentose sugar is the backbone. Okay, pentose sugar is the backbone. Then you have phosphoric acid, okay, or you can write H3PO4 phosphate group, okay. Then you have nitrogen containing bases, okay, nitrogen containing heterocyclic structures, okay, heterocyclic compounds. So these are the what? These are the composition of nucleic acid. So remember this, very important. Pentose sugar, phosphate group, okay, and nitrogen containing heterocyclic compounds, or you can say nitrogenous bases. Okay. So these are the compositions of your nucleic acid. Is it clear? Is it clear? Okay. Next, we are going to study in detail about all. So first we are going to study about pentose sugar. Okay, we are studying about pentose sugar. So there is DNA and there is RNA. Okay, so in DNA, so in RNA, the sugar which is present is, remember this, please note it down. The sugar which is present is beta D ribose. Okay, the sugar, pentose sugars which is present in RNA is beta D ribose. And in DNA, which sugar is present? It is beta D. Okay, it is beta D deoxyribose. Okay, it is beta D deoxyribose. Okay, so this is the sugar. Now let me write the structure. So beta D. So remember this. Please note it down. Very important. Please note it down. OH, H, H. CH2, OH, and H. So how many carbons? So 1, 2, 3, 4, and 5. So this is your beta D2 deoxyribose. So at 2 position, you can see there is no oxygen is missing, right? So it is deoxy. Now here in ribose, okay, let us write the structure of ribose. So this is OH, H, this is H, OH, this is H, OH, this is CH2, H. Okay, so 1, 2, 3, 4, and 5. So this is our beta D ribose. Understood? Which is the pentose sugar. So please note it down. Very important for your competitive point of view as well as board point of view. Okay, they may ask you for 1 mark question also. Which is the sugar present in RNA? Which is the sugar present in DNA? So with different options. Is it clear? Should I clear this? Did you note down the structures and the name? Okay. So let us go for the next one. So we have discussed about the pentose sugar. Next, we are going to discuss about the nitrogenous bases. Okay, nitrogenous bases. So which are the nitrogenous bases? So DNA, okay, consists of 4 nitrogenous bases, that is adenine, okay, then guanine, then cytosine, and thymine. Okay, so how they are represented? This is represented by A, G, C, and T. So, what does your this consist of? DNA consists of 4 nitrogenous bases: adenine, guanine, cytosine, and thymine. Okay, so RNA. What does RNA consist of? RNA consists of again 4 bases. Okay, first one is adenine. Second one is guanine. Third one is cytosine. Fourth one is uracil. Okay, so this is represented by A, G, C, and U. So instead of in DNA, thymine is there. In RNA, what is there? Uracil is there. Okay, so these are the 4 different nitrogenous bases. Is it clear? Is it clear? So in DNA, adenine, cytosine, guanine, and thymine is there. And in RNA, adenine, guanine, cytosine, and uracil is there. Okay, so remember this, very important. Very important. Uracil is present in RNA and not in DNA. Okay, so these are the 2 important things, okay, about nitrogenous bases. Is it clear? Okay. I hope you have noted it down, right? Okay. Next is structure of nucleic acid. Okay. So how the nucleic acid is formed? Uh, so base, okay. Our base, it is attached to the sugar. So here you suppose, so this is the base, nitrogenous base, okay. And this is connected to phosphate, okay. So that is to a pentose sugar, H, OH, HOH, again base and H. So this is your nucleotide, okay. And this is your HOH, this is your HOH, this is your CH2OH and H. So this is your nucleoside. This is your nucleoside. So a unit is formed by the attachment of your base, okay, to the sugar, and this is called as nucleoside. Okay, and when this nucleoside, when it is linked to a phosphate group, When it is linked to a phosphate group, then then it is called as a nucleotide. So this is the structure of nucleic acid. This nucleotides, they are joined by a bond which is called as the phosphodiester bond. So remember this. So which is the linkage present in the nucleotides? That is a phosphodiester linkage is present. Okay, the phosphodiester linkage is present. Okay, so this is about your nucleoside and nucleotide. Now, next is the structure of DNA and RNA. Okay, so DNA, how it is? DNA is having a double stranded helix structure. Double stranded means what? Now you are like this. Okay, like this. So like this is the structure of your DNA. So we have seen helix means it was singly coiled like this. Okay, DNA has double stranded. So from here also, from this side also. So DNA is having a double strand helix structure, and this was given by Watson and Crick. Okay, Watson and Crick were the scientists who gave the structure of DNA. And these the two nucleic acid chains, they are held by so hydrogen bond. Okay, so the hydrogen bond is present between these two nucleic acids. Okay, and the pairs so between these pairs, now suppose how the pairing will take place, how the pairing will takes place. Adenine, okay, adenine, if adenine is present here, it will pair up with thymine, okay, with double bond. And if cytosine is there, cytosine will bond with your guanine with triple bond, like this. Okay, so like this the bond formation takes place. So this is, so what happens that these two strands, now if your 5 dash starts, then your 3 dash, here 5 dash, then here 3 dash, like this. So the strands are what? They are complementary to each other. They are complementary to each other. And what is present between the two pairs nitrogenous base? Hydrogen bonds. Okay, so hydrogen bonds are responsible for the to hold the nitrogenous bases. Okay, and they will form a specific pairs of bases are present between these two strands. So adenine will pair up with thymine and cytosine will pair up with guanine. And RNA, when we are talking about RNA, RNA is a single strand. RNA is a single strand of polypeptide chain. So like this, okay, it is a single strand of polypeptide chain. So what does RNA consist of? RNA consists of mRNA, okay, it consists of tRNA and it consists of rRNA. Okay, so these are the, this is about your DNA and RNA. So only this much is there for your syllabus. So this much only you need to know. Okay, is it clear? Is it clear up to here? So what is, so we have discussed the composition of nucleic acids. Okay. So composition of nucleic acid, you need to know the which are the sugar present. Okay. Then which is the which are the nitrogenous bases present in DNA and RNA. Then we have discussed about the structure of nucleic acid, right? How is the double stranded helix structure of DNA? Okay. Then so this is about the DNA and RNA. Any doubts up to here? Yes. So in this chapter, what all we have discussed? We have discussed about the carbohydrates. We have discussed about the proteins, right? We have discussed about the enzymes. We have discussed about the vitamins. We have discussed about the nucleic acids. Okay. So these all things are discussed in this particular chapter. And we have discussed about hormones also, right? So these are the important biomolecules which are important. Okay, in our this what for functioning of the human body. Okay, so for the growth and maintenance of the human body. Is it clear? Is it clear? Any doubts up to here? The weightage for this chapter is around, just a second. Okay. This particular chapter is for 9 marks. Okay. This particular chapter is for 9 marks. Fine. So the last unit it was biomolecules. So we have completed all the 10 chapters for your board syllabus. Okay. So any doubts? Okay, fine. So this completes your entire syllabus of PU2 chemistry. Okay. So if you have any doubts, I think you are there in the group, so you can ask your doubts there. Okay. So we will be there anytime available for you all. Okay. So if you have any doubts, you can reach us anytime. Okay. Fine. So no doubts now. Okay, fine. So I shall end the session here. Okay. So we will meet soon in the next classes for CET. Okay. So CET classes will be soon. So that time we will all meet up. Okay. Fine. So thank you. Yes. Thank you. Yes. Thank you, dear students. Okay. So we'll meet soon. Okay. Bye.